Okay, thank you, Jeremy, for the invitation and for the nice presentation. So we're going to go from sharks to viral vectors, and I will give you basically an overview of the challenges of basically developing therapeutics based for using the concept of gene therapy. I'm going to start with some slides that are kind of introductory, because probably you're not super familiar with gene therapy. What is important to know is that what we're talking about is a complex multi-component therapeutic. In fact, in a gene transfer vector, we have a vector which can be a virus or a recombinant virus derived from the more or less engineered natural variant of a virus infecting humans or other animals. There's a transgene, so a coding DNA expressing a therapeutic entity, and there's a target tissue. In fact, all these three elements can concord to determine the immunogenicity of gene transfer therapeutic. The toxicities can be related to both the vector of the transgene. I'll show you some data mainly focusing on the vector. And finally, the target tissue can greatly act as determinant of the outcome of gene transfer in terms of immunogenicity. Two basic approaches. Today we will talk about in vivo gene transfer, where the vector is delivered directly into an organism. Of course, there's the other side of the story where autologous stem cells are taken out of an organism, then transformed with the virus and put them back. For in vivo gene transfer, AAV vectors or adeno-associated viral vectors are the vector of choice at the moment. These vectors are derived from the wild-type virus, which belongs to the parvovirus family. It's a dependent virus, so it needs a helper virus for replication and is non-pathogenic. And these characteristics really started the whole story of this vector. Recombinant AAV vectors are derived from this virus by simply replacing all the viral coding genes with an expression cassette of the gene of interest, which then is flanked by two inverted terminal repeats, the only viral sequences that are present in AAV vectors that are needed for packaging. And you can package your transgene in several flavors, so different serotypes of AAV depending on your target tissue. One important point about these AAV vectors is that they can be produced in large quantity at a good level of purity, supporting clinical studies. And another very important point about AAV vectors is that once they are introduced into a cell, they infect a cell and they transfer the transgene, they can drive long-term expression of the donated gene. This is the example of muscle collected from a Hemophilia B patient 10 years after gene transfer, showing that you can still retrieve the viral genomes and the vector is still expressing, and you can detect the product of the donated gene at the protein and RNA level. And there's a considerable experience in humans with these vectors. This is just an old list. In fact, the list now is much longer of trials that have been conducted in humans using these vectors for both inherited and metabolic and acquired diseases. And perhaps some of the most recent highlights and most important results are here. The first one with AAV vectors was the correction of Hemophilia B. This was studied that was conducted, sponsored by UCL and by St. Jude Children's Research Hospital in the States. And finally, last year, we had the approval of the first gene therapy product. So I'm going to take hemophilia as a model to discuss the issues that were encountered with AAV vectors. I don't really need to go over this slide. We heard about it several times. In fact, the only point that is important is that to achieve phenotypic correction of hemophilia, you don't need too much expression of a transgene, or you don't need too much protein. In fact, you can convert a severe bleeding phenotype to moderate by simply increase the levels of clotting factors to about 5% of normal. This was a very important point that really triggered the initial interest in the disease of the gene therapy community. So we really don't need much of expression of a gene to correct the disease. Several groups showed also that it's possible to express these genes in different tissues, including muscle and liver, and you can really produce functional clotting factors. You don't need a precise regulation of the transgene, and also in the case of hemophilia B, the actual cDNA that encoded for factor 9 is small enough that can fit in these AAV vectors. Finally, animal models were available. And so this point really started the whole story of hemophilia, as I told you, and this is just one of the results that were obtained. This is the case of hemophilia B dogs. In these dogs, we showed that after delivery, a single delivery of an AAV vector encoding for factor 9, it was possible to correct the disease. And what is important is that after over 10 years, these dogs are still expressing the donated gene at the same levels that we're doing, let's say, after reaching a plateau. So these studies, and several others, of course, led to the initiation of the first clinical study in which an AAV vector was introduced into the liver of severe hemophilia B patients. This was an open-label dose escalation study. This is very old data. Initially, 3 doses were tested, 2 of which were subtherapeutic, but this was because this was a first-in-human study, and so we were required to start at low doses of vector. What happened in this study was that in the first subject, in whom the vector was given at the high dose, in this case was 2 × 10⁻12 vector genomes per kilogram of weight, expression of the donated gene was observed. You can show here the red line. You can see here the red line. But what was different from this subject to what was observed in experimental animal models was that the expression was then lost. And these lots of transgene expression started around week 4 following vector delivery, and was followed also by an increase in liver enzymes. So there was a liver damage and a loss of expression of the transgene, which was observed in this subject, and the same increase in liver enzyme was observed also in another subject. So we did a lot of studies at the time to try to understand why this was happening, and we really focused on the issue of immunogenicity of our vector, and in particular of the two antigens that were associated with the delivery of this vector, which were the capsid, non-replicating capsid, and the factor 9 transgene that was encoded by this vector. And this is really a summary slide of many studies. What is key is this panel showing that we were able to demonstrate that after vector delivery, there was an expansion of a population of capsid-specific CD8-positive T cells. As you can see here, we monitored the cells in PBNCs using pentamer. There was an expansion and contraction, and more or less this overlapped the increase in liver enzymes. We performed several other studies. Of course, we excluded that this immune response was directed against the factor 9 transgene, and in fact, this subject really continued to respond to the normal enzyme replacement therapy. We also demonstrated that the cells were cytolytic and they were highly cross-reactive, so they were able to recognize different AAV capsid, but this was also not surprising because there's a high degree of conservation of the sequence across different AAV serotypes. And so this is essentially the model that we came up, and I will really go briefly into some of the studies that we performed to validate this model. But basically, simply is the model of a viral infection. The only difference is that the vector the virus in this case is a vector. So it's a recombinant vector. And so basically the vector enters the cell, in this case the hepatocytes, and then is processed and presented through the proteasome pathway and onto class 1, where it's recognized by CD8 positive T cells. And I don't really have the time to show you this data, but we did show that it was possible to detect antigen at the surface of transduced hepatocytes. We showed that the proteasome pathway was key to the antigen presentation. And we also did several studies looking at the counterpart of antigen presentation, which are the T cells. In particular, this is a study that is still unpublished. We look at splenocytes collected from humans, and we show that if you look in children below the age of 5, the reactivity against the capsid is fairly low, but then in older individuals, the reactivity goes very high, up to 80% of the population results positive for T cell to the capsid, directed to the capsid. But this is not surprising because humans are natural hosts for AAV. We also developed tools using the splenocytes to map epitopes triggering MHC class 1 epitope triggering these responses. In this case, we perform a single peptide screening using an ELISpot, interferon gamma ELISpot assay of splenocytes from healthy individuals. And we were able to map a series, with this approach, a series of peptides binding to different HLA types. So we were able to determine some sort of a collection of epitopes that we could use for immunomonitoring. We also look at the correlation between, of course, the reactivity on the T cell assay and the basically neutralizing antibodies, showing that there was absolutely no correlation. And this was done in the laboratory at Genethon. However, we also developed the same data set in Philadelphia. And finally, this is another a set of slides that I wanted to introduce here. We also developed an assay to basically screen the T cell reactivity for large genes. And this was really something that we did because we also are very sensitive to T cell responses to the transgene product, and at Genethon we are very interested in muscular dystrophy. So if we wanted to screen for T cell reactivity against dystrophin, we would need basically a peptide library of about 730 peptides, and this is really a problem. And so what we did, we developed a modified ELISpot assay where, as a source of antigen, we use a lentiviral vector, which is used to infect PBMCs, which are then screened in the ELISpot. And these are some of the results which are published. Essentially, you can see how these restimulated PBMCs that can be used both in ELISpot, which are the gray bars, and intracellular cytokine staining, and we can effectively detect T cell activity against an antigen with this approach. I just wanted to add this assay just because it's something that was a big challenge for us to develop. Anyway, so T cell activity against the AAV capsid is an important problem in gene transfer, and we spent probably, since the initial observation, a good over 5 years trying to think about possible solutions to this problem. You see a list here, and I will show you some of the data from the London trial that I mentioned at the beginning of my talk, just to show you what was done to address this problem, which was essentially to use a vector highly efficient in targeting the liver, which would allow, in theory, to achieve therapeutic efficacy with a lower total antigen load, and also use immunosuppression. This is the outline of the trial, very similar to the one done at the beginning, except, as I say, a very efficient vector, and the fact that in case of an increase in liver enzymes, the protocols was written such as there would have been the administration of a course of steroids. And these are some of the data. I would not go into the detail except to say that there was expression of the transgene, long-term expression, and the same problem of immunogenicity was encountered as shown here. So this is the data of the first subject from the Highdose cohort, where in red again is the transgene expression. As you can see here, this after vector delivery, this subject was expressing factor 9, about 7% of normal, and then all of a sudden the transgene expression went down to 3%, and at the same time there was an increase in liver enzymes. So at this time there was a course of prednisolone was started, high dose, so immunosuppression was started, and this really led to the resolution of the transaminitis, as you can see here, even after the steroids were discontinued. What is also important is that also in this subject, this is a interferon gamma ELISpot assay for using peptides against the AAV capsid. We did observe an increase in T-cell reactivity around the same time of the increase in liver enzyme and loss of transgene expression. This is just showing that some of the epitopes that we predicted were contained in the pool that had that showed the highest T-cell activity. And this is basically the summary of the overall study. You can look at the upper table, showing that essentially with increasing doses of vector, there's an increasing T-cell activity. So at the intermediate dose, at the low dose, there was no T-cell activity detected by ELISpot. As the dose increased to the mid dose, all the subjects had T-cell activity, but none of the subjects had an increase in liver enzymes. And finally, at the highest dose of vector, all the subjects had T-cell activity to the capsid, but only 4 out of 6 required an intervention with steroids. So the summary of the first part of this presentation about T cells is that, okay, T cell responses can be triggered by the administration of AAV vectors. It's now clear that this is the case, and it's not only a result coming from liver gene transfer, but it's coming also from other AAV trials. The magnitude of these responses seems to correlate with the total antigen dose, so the vector dose. There is some sort of a threshold above which this response becomes clinically relevant. It is possible to block the response using steroids. However, at the moment, what is important is that we don't really know why some subjects will require steroids and some subjects not. And so what is really driving the immunogenicity of gene transfer in this context? I'm just going to quickly go through a few slides. It's all published work. We also tried to use regulatory T-cell epitope to modulate responses, T-cell responses to the AAV capsid. Essentially, we showed that using these peptides, you can expand Tregs using human PBNCs. You can suppress CTL responses against the AAV capsid in vitro. So these are cells, TCTLs, incubated with these regulatory T-cell epitopes, and so you don't see killing of target cells. And you can do the same in vivo if you create fusion proteins of capsid with, so the offending antigen with this regulatory T-cell epitope. You can decrease CD8 T-cell activity in mice and increase the frequency of CD4 positive, 25 high, phospho-3 positive T-cell in spleen. So I'm going quickly to the next chapter, which is humoral immunity to AAV. It's another big issue in gene transfer. Essentially, in this room, if we do a screening of sera from people in this room, probably the majority of us will have antibodies against AAV serotype 2. And this is a real issue because If we have that, we cannot do gene transfer. The vector is simply cleared before it reaches the target cell. This is, for instance, a survey of prevalence of AAV antibodies in the general population that was done at Genethon, showing that, as you can see, the prevalence is pretty high across several serotypes, except maybe AAV5. What is important to see is that the cutoff for positivity chosen here is pretty high as well, is 1 to 20. And in fact, if you look at animal data, you can see how a titer much lower than 1 to 20, 1 to 5, can completely prevent transduction of the liver with an AAV vector given through the circulation. So this is a big problem, and even bigger is the problem of vector re-administration, if needed. And this is the case of this table is some data coming from patients 9 or 10 years, up to 10 years after gene transfer, showing that if you give a vector to a subject, this person will develop very high titers to the vector itself, which will persist for a long time. And in this case, you can see how this subject receives an AAV2 vector, but the antibodies are highly cross-reactive against also AAV8. So this is a very important problem. There are several strategies that have been tested to overcome this limitation. The bottom line is that yes, they work to some extent, but at high titers they don't really work. So we ask ourselves how we could design better strategies to overcome this limitation. And to do that, we also wanted to develop a mouse model that would be suitable to really do these studies. And to do that, we basically took mice, regular C57 black 6 mice, and we injected IP with intravenous immunoglobulin, which typically has very high titers against the AAV of all serotypes, really, being a large pool of human IgG. So 24 hours after, we basically measure antibodies in these mice, and we showed that very reliably we were able to immunize these mice at the titer we wanted. So from naive, we were able to get very high titers based on the dose, and this was really very reliably achieved. And so after that, we used these mice in an experiment where we basically gave AAV vector alone or AAV vector mixed in essentially capsid that did not contain any genome. We call them empty capsids. These empty capsids are basically a byproduct of production. We don't know what to do with it, and usually we discard them. But this time we wanted to see whether this empty capsid really had an effect on the outcome of gene transfer. And so starting from mice with low but detectable titer, you can see how naive mice, we gave a vector and we see expression. We call this 100%. Then if we immunize the mice with this IVIG, we block the vector transduction. But then if we mix our vector increasing amounts of this extra capsid, we can really rescue the transduction. And so we went on and show different antibody levels. You see 1 to 3, 1 to 10, high titers and very high titers, that we were able to overcome the limitation of antibodies to AAV by simply mixing the vector with a dummy vector, a decoy vector, which would really absorb all the antibodies and allow for transduction of liver. The only problem is that if we add too many decoys, 1000x the dose here, we can actually inhibit transduction. And so, okay, this is just is the validation of the studies in monkeys. This is the concept that basically is very simple. We have antibodies against AAV, we have an AAV vector, and then we have this with capsid decoys. And if we outnumber our vector with these decoys, we can rescue transduction. And what is important is that gene therapy, for once, is a simpler the issue of antibodies is a little simpler in gene therapy compared to protein therapeutics, because we're talking about a one single intervention, so we need to go in only once, conceptually. So we also, and this is I promise my last 2 slides, went on and further refined our approach by basically taking the wild-type virus and modified the virus by killing it. So we produced non-infectious AAV particles by basically mutagenizing the receptor of the virus. And this is just showing that if we make an AAV mutant vector with this approach, it just doesn't transduce cells, is not able to enter cells. And with this maneuver, we created essentially non-infectious decoys that you can see here in red, that can be used to rescue the transduction of AAV vectors, and we can mix them with the vectors even at high proportions, and they don't compete for receptor binding. What is also important is that we can use the same decoy to protect different AAV serotypes from neutralization by antibodies. Now, I don't have the time to show you more data, but we did extensive studies around the safety of the approach and biodistribution and so on. So I'm going to close this by just concluding the second part of the talk. Neutralizing antibodies are perhaps the biggest concern at the moment in terms of for what it concerns the gene therapy, especially when the vector is given systemically. We can see how T cells, we can block them with steroids, but with antibody we don't have a good solution for that. The problem is big because it forces to exclude 1/3 to 2/3 of the population from gene transfer trials. It is very important to have a very sensitive assay, because at the moment we don't have that. I didn't really show you the data, but we don't have it. It's also important to know that, okay, the only problem with it is the lack of efficacy. There's no toxicity associated with antibodies. So finally, I cannot rush through this part of the presentation, but I just wanted to say that we are getting there as far as solution is concerned, and probably this will be a combination of strategies, which may involve the use of this capsid decoy, maybe associated with other strategies like pharmacological immunosuppression or plasmapheresis. Finally, this is the long list of people involved in these studies. Of course, the people at the Children's Hospital of Philadelphia, the group led by Katie High, my group now at the UPMC Geneton, and all the other people involved in immunology studies and the clinical studies. Thank you.
Great, thanks very much, Federico. I just want to start maybe asking us a little bit about the question of anti-factor IX immune responses. Is that something which you've seen any issues with, particularly in patients which haven't had the expression of factor IX at all? So when that's expressed, do you see any immune responses to the actual transgene itself?
No, so far there was not such an issue in any of the patients enrolled in. Oh no, that's fine. Okay. In the study, so all the patients were also pre-screened in a way that basically they were pre-exposed to recombinant or plasma-derived factor IX and didn't have an history of inhibitor formation. However, it's also true that the approach should be safe for what it concerns immunogenicity of the transgene, especially the liver-directed gene transfer, based on preclinical data showing that if you express a gene in the liver, you induce tolerance to the gene. And in fact, in dogs, in the case of Hemophilia A, this approach is being used by the lab in Philadelphia to eradicate inhibitor. So it seems safe.
Do all of the AAV serotypes exhibit tropism for for liver, or can you target other specific?
Yes. So depends. If you give a vector systemically, a lot of the vector will end up in the liver and in lungs, mostly liver. But there are serotypes that are better doing that, and serotypes that are worse. So, and also people have engineered some serotypes by basically modifying some of the exposed loops in the protein that are basically interacting with the receptors or the cellular receptors, and they were able to detarget liver and, for instance, move the target into muscle preferentially.
Thanks. Really interested to see your decoy approach. Do you run into problems with getting those accepted by the regulators to go into your patients?
Sorry, I didn't.
So do you have a regulatory issue that you're putting in essentially a different kind of drug as opposed to your AAV?
Good question. So we don't know that because we haven't tried yet the use of mutant decoys. The use of decoys, however, has been approved for a phase 1/2 and is being already given to patients. So the approach in that case is, I didn't really have the time to explain, but basically when you produce a vector, you will have a considerable amount of empty capsids, these decoys, that you can use as decoys. So what we did, we prepared the two separately, and we mix them at the time of dosing in a proportion of 1:4, and that was approved. But they're the same capsid.
In your decoy capsid treatments, do you ever see evidence, this is in people who already have titers, right, to AAV?
They're screened for having low titers.
Do you see any evidence of boosting?
In people, I don't have the data yet, because it's really ongoing now.
You're basically immunizing them again?
Well, we did some work, and actually is part of the supplementary data of the published manuscript. So we didn't really see a boosting effect in mice and monkeys. Probably because the titers, after you do an vector administration procedure, they shoot really high, and you don't really see a boosting effect.
I'm sorry, I didn't follow that. You're saying when you give them this,
to mice and monkeys, we try to give vector alone, or vector and decoys, or decoys only, and the titers look essentially the same, very high.
But they go up.
Very high, yes.
Extremely high.
Yeah.
So is that a cautionary note for repeatedly dosing people like this?
Well, from issue of it's a good question. From an issue of, let's say you're making antibody antigen complexes, which you can run into trouble. We did studies in monkeys in which we really look at all the I mean, more studies probably are needed, but we did some study and we didn't see any evidence of toxicity. It's also true that the amount of protein that we're talking about in the case of gene transfer, it's low. So if you take into consideration that the highest dose given to an individual is probably in the order of 10 to the 14 total particle, that amount is about 0.6 milligram of protein. So it's lower than what you would discuss in ITI, for instance, for immunotolerance induction in hemophilia, where you need a lot of administration. So there you only have one-time administration of a low amount of protein.
Okay.
Thank you.